Okay, hello, welcome back. This is the second super resolution microscopy lecture that I'm giving, and in this lecture we've already covered, in lecture 1 we covered the diffraction limit, SIM microscopy and STED microscopy, and in this talk we're going to be covering single molecule localization microscopy and more advanced techniques, where the field's going, what's the state of the art, what's cutting edge. So, single molecule localization microscopy, also called SMLM, and there are going to be some other acronyms which we'll kind of grapple with in a little bit. So, to begin with, let's think, what does an image of a single fluorescence molecule look like? Okay, so here is a real image of a real molecule. This is an image of one molecule of Fluor 647. Okay, one molecule that I've imaged. Cool. Boring, but cool. So I want you to look at that image and I want you to think, do I know where the centre of that molecule is? And how confident am I about where the centre of that molecule is? So if I look at this, I'm like, well, I'm pretty sure the centre of the molecule is somewhere around here. You know, it's not going to be over here, it's not going to be over here, not even going to be here really, maybe here, but most likely here. So we can already infer higher resolution information from this image of a single molecule than is actually in the image. We can pinpoint that centre kind of mentally better than we can see in the image. And a lot of the time, if you can do something mentally, you can do it computationally. So what I can actually do is fit a two-dimensional Gaussian to find the centre of this molecule very accurately. So this is just exactly the same information, where intensity is on the z-axis here and xy just here, and this is a fit to the intensity of this molecule. And from the parameters of that fit, I can say, okay, cool, the centre's there, and I can relate that back to my original image to find the centre. Okay, so if we have an image of one single molecule, then we can pretty accurately work out where the centre is pretty easily. You can do a Gaussian fit. And the quality of this Gaussian fit, so how kind of confident we are in the fit, can then be related to our confidence in where that centre is. So, you know, this kind of represents the error in our localization, basically. So if we can fit a Gaussian function to an image of a single molecule, we can obtain the centre and essentially a confidence interval on where that centre actually is. So something like this. So we can turn our raw data into that higher resolution. Great. So now if you think about a wide field fluorescence microscopy image, we've got, for example, this is some microtubules labelled with Fluor 647. So again, same dye as we looked at before. And all this image is lots and lots and lots of those single molecule images on top of each other. Okay, so it's one image containing really complex spatial information. What we do in single molecule localization microscopy is we try to find those single molecules again. And to do that, instead of taking one image where all of our molecules are emitting fluorescence at the same time, we instead take a really large number of images where a very small number of fluorescent molecules are emitting in each frame. So we go from one image containing very complex spatial information to a large number of images, each containing much simpler spatial information. And you can see this looks like, this is called kind of blinking, because it really looks like blinking. The way this is done, and we'll talk about how we generate the blinking, don't worry, is we actually get the fluorescent molecules turning on and off. And if we look between the difference between these two images, in each of these individual blinking images, I reckon you could even just, you know, if you had a pen and paper, just pinpoint the centre of each of those molecules and start to build up a really high resolution image of our data. So we don't do it with a pen and paper, obviously, we do it with this Gaussian fitting method, and we fit Gaussians all over the image to find single molecules, and then we render that as an image. So this is the image building up as it finds more and more molecules. Cool. And you can see we've got a much higher resolution here than we did before. So to kind of very briefly summarise single molecule localization microscopy, we spread the fluorescence out in time so we can see single molecules, and then we can accurately localize the center of each of those single molecules and build a map of where they are inside the sample. Great. So as you can imagine, one of the most important bits of this whole technique is how do we actually achieve that blinking? You know, if we go to the microscope and we put our sample in the microscope and we acquire a long time series, it doesn't just magically start blinking. To do this, you need to be able to have a fluorescent labeling system with an on state and an off state. Okay, so our on state is fluorescence and emitting light, and we can see it. Our off state is dark. Okay, and we need to be able to at least transition in one direction between these. And those are this kind of off switching rate and on switching rate. So determining how fast you go between these fluorescent on states, non-fluorescent off states. And the difference between a lot of single molecule localization techniques comes from how you generate the blinking. So one of these is called PALM, which is photoactivatable localization microscopy. And in PALM, you achieve the blinking by using fluorescent proteins, which have a dark state and a light state, or some kind of on state and off state that you can transition between. Another method is STORM, stochastic optical reconstruction microscopy. And in this case, you don't have fluorescent proteins with an on state and an off state. You have organic dyes, such as the fluor dyes, that can go between off states and on states. And the final kind of popular way of achieving blinking is DNA paint, which is points accumulation for imaging in nanoscale topography. And in this case, this uses melting of DNA to bring short labelled oligonucleotides closer and further away to your sample. So that's your on state and your off state, the distance from your DNA molecule to the structure of interest. So we're going to go through each of these individually and just get a flavour for what they're like. So how do we achieve blinking in PALM? This is where we use fluorescent proteins. So the first method is using photoactivatable fluorescent proteins, such as photoactivatable GFP. So photoactivatable GFP is kind of a broken GFP. So if you just label your sample with photoactivatable GFP, you've got this kind of broken chromophore in the middle of your fluorophore, and it's dark, okay? So if I try and image it like normal GFP, I won't get any fluorescence out. So that's my off state. If, however, I shine some ultraviolet light, some activation light onto my GFP, so shine some on, some of my molecules will randomly undergo this rearrangement of hydrogen bonds to activate the chromophore. Okay, so if I apply UV activation, then some of my GFP molecules will become active, so it'll turn into kind of a functional GFP, which can then be excited and emit fluorescence. Great. And then after a while, this can actually go back to the dark state, or the on state can be permanently bleached. So we can have these on and off transitions. And so you can imagine if you're imaging, you have UV activation at a level that continually puts fluorophores into the on state, and your excitation, which essentially gets your fluorescence and then converts back to the off state. So it's this balancing act of having your activation light and your excitation light to get your blinking rate nice, so you get individual molecules. Another class of fluorescent proteins that are used in PALM are photoswitchable fluorescent proteins, for example, Dendra-2. So in Dendra-2, the off state is quite a bad green fluorescent protein. So if Dendra-2 is in its off state, you try and image it with 490 nanometer excitation, and you'll get some really kind of bad fluorescence. You'll get low intensity fluorescence out. But the other thing that happens when you excite this off state of Dendra-2 is, again, it can cause a change in the chromophore structure in the center. So in this case, carbon-carbon double bond formation. And in Dendra-2, illuminating with 490 nanometers can actually turn this green fluorophore into a red fluorophore. Okay, so in that case, if we then also put on some excitation light at 561 nanometers, then we'll get lots of red fluorescence. So in Palm, our blinking is mediated by the laser intensities and the wavelengths. In our photoactivatable proteins, we'll have UV and our excitation light, balancing those to get the right ratio of on state to off state. And with our photoswitchable fluorescent proteins, we'll have our essential conversion light, which makes it into the red state, simultaneously with our excitation light, which generates the fluorescence that we actually detect. So that's how Palm works. STORM is slightly different. So this is using small organic fluorophores instead of fluorescent proteins. So the on state in STORM is basically the same state that we use in all conventional fluorescence microscopy, where we have our excitation and our emission. And we're just aiming to go around this cycle lots of times, generate quite a lot of emission, image our structure. Now, these aren't the only states that a fluorescent molecule has. This is just a representation of the singlet state. There's also a state called the triplet state in molecules, and technically, because of quantum mechanics, transitions between the singlet and triplet states are forbidden. It's a quantum mechanical, forbidden transition. But, again, because quantum mechanics is wacky, you can break that law, you can break that forbidden transition if you try hard enough. What does that mean, if we try hard enough? Well, let's say we really increase the intensity of our excitation. What does that mean for this molecule? Well, as soon as it'll get up to the excited state, it'll emit fluorescence, and then it'll immediately be driven back into the excited state. So when we really strongly increase the intensity of our excitation, our molecules end up spending way longer in our excited state than they would with a lower intensity. And if you're in the excited state, there's a small but non-negligible probability that you'll slip into the triplet state. Okay, so you can't go there from the ground state, you can only go there from the excited state. It's a very low probability process, and you increase the probability by essentially making sure your molecule's up in the excited state as much as possible. Okay, so we go into the triplet state. Cool, so this will happen randomly, it's low probability, and occasionally you'll just fall into there as a molecule. If you're in the triplet state, you can't do fluorescence, so you're dark, okay? And that's great, right? We've got a small number of on molecules, and we end up with quite a large molecule of off-state molecules because, again, we can't do fluorescence in the triplet state, and the triplet state has a much longer lifetime compared to the singlet state. So once it's in the triplet state, even though it's quite a low probability this happens, it stays there for ages, okay? And that can be up to seconds. You know, one fluorescence cycle takes a few nanoseconds. The triplet state lifetime can be in excess of seconds. So that's how the blinking is generated in STORM. We knock our molecules into the triplet state, and from here they can undergo some more transitions. So within the triplet state, they can then go to a radical state, and from that radical state they can actually undergo permanent bleaching. You've broken your molecule. Alternatively, you can try and bring the molecule back to the singlet state, and that's via chemically mediated transitions, so normally with buffering. So also you can mediate this kind of transition back from the triplet state to the singlet state with UV light. So again, technically transitions between singlet state and triplet state are forbidden, but you can engineer conditions where actually they're slightly more likely than you'd expect them to be, okay? So blinking in STORM is mediated by several things. Firstly, the excitation intensity, so this is the thing that shoves stuff over into our off state, our triplet state. Our buffering, which are the chemically mediated transitions, which can bring things back from the off state to the on state, so fluorescence again, and any UV light that you put on the sample, because that can also help recovery from the off state to the on state. So again, if you have the right combination of excitation intensity, correct buffering conditions, and UV light illumination, then you can actually quite nicely fine-tune the blinking statistics of your molecule. Okay, the final way of generating blinking is paint, DNA paint. So in DNA paint, this is our off state. So instead of labelling our target molecule that we want to image with a primary antibody that's, say, got a fluorescent dye conjugated to it, or is fused to a fluorescent protein, we do antibody labelling, but instead of a fluorophore, you actually put a short strand of DNA called the docking strand. Okay, so it's a single strand of DNA. That's how we label our sample, apart from, as you can see, there's no label there. You then also put into your sample imager DNA strands. Now this is the complementary sequence to our docking DNA strand with a fluorophore attached to it. And the idea is you can design these DNA strands in such a way that you'll get random binding and unbinding. You can really exploit the kind of melting of DNA to do this, kind of to bring your fluorescent molecule closer and then further away from your target. And so this happens randomly, right? Every so often you'll have your DNA strand diffusing by, and it'll bind to your docking strand, and then that's your on state. It's emitting fluorescence. After a while, because of the sequence and the base content of your DNA strand, that will unmelt and diffuse away. And you get this on-off of image DNA strands that are in your kind of imaging solution, transiently binding and unbinding to your target of interest. And so the things that mediate your blinking probability, your blinking rate in this case, are the sequence of the DNA strand and also the concentration of imager strands. So put more and more and more imager strands in, the more likely it is that one will come close enough to your target strand to bind and make a blink, basically. But basically, if you're the algorithm, you don't care how it's generated, okay? So if you're the part of the process, if you're the computational process that's looking for single molecules, normally you're pretty agnostic to how it was generated. So the key thing is that no matter how you actually get your blinking, at the end of the day, it always looks pretty similar. It always looks like little short flashes of light in your sample. So regardless of how you label your sample, it's normally analysed in exactly the same way. The only thing that makes a real difference is how dense your blinking is. So if you have lots of molecules overlapping, so more towards this central regime, things can get difficult. So if your algorithm can't actually accurately discern what's one molecule or two molecules overlapping, you do start to get problems. But ideally you'll have nice sparse blinking where it's really easy to see individual fluorescent molecules blinking, however you generate that. So the key features of single molecule localization microscopy are it's a wide field microscopy technique, and the optics are pretty simple. So for STEM we need to put in some kind of grating or a pattern generator to make the high frequency pattern for the wire interference. Instead, you need to put in a second laser beam with optics to make that donut. In single molecule localization microscopy, you just need a wide field microscope with high intensity lasers, so we can, for Storm and Palm, make sure that we're mediating those transitions correctly. And the spatial resolution of single molecule localization microscopy is also technically unlimited. The only thing that limits your resolution is how good your fit is to a single molecule. So again, if you had an infinitely bright blink that you could fit really, really, really accurately, you'd have infinitely high resolution. So what are some applications of single molecule localization microscopy? This is one of the coolest things that has been found using super resolution microscopy. And this is the kind of cytoskeletal substructure of neurons. And again, this can be done in 3 dimensions. I won't talk about how you go into 3 dimensions with single molecule localization microscopy, but it can be done, and I'll provide some reviews if you want to learn more about that. But this is 3-dimensional single molecule localization microscopy, STORM in this case. And what was seen was that if you did storm on various cytoskeletal components such as actin, spectrin and adenosine, what you see is this really beautiful striped pattern along axons in this very periodic manner. And looking at different proteins, you see they actually kind of intercalate. There's this very regular, repeatable spacing and this kind of pattern that forms along axons. And from this, they can actually make a model showing how cytoskeleton is organised within the axon. Each of these kind of actin rings is about 190 nanometers separated, so they couldn't be seen with conventional microscopy. And they think this serves like a really important role in compartmentalization of things like sodium channels to specific regions of axons. So this is really cool. This is like one of the most beautiful super resolution studies, in my opinion. It's a real structure discovery. More recently, this is an example of STORM being used for kind of structure discovery in virology. So this is vaccinia virus. It's a very, very large virus. It's about 300 nanometers in size, but in kind of the context of resolution, problematic, right? And what was seen here was that different, when you label different membrane proteins in vaccinia with LEX647 or STORM dyes and image them, you can actually see that there's some kind of organisation of different proteins within the virus itself, which is in the virus membrane. For example, these proteins here polarised to the tips of the membrane, and this played quite an important role in its ability to fuse to the host cell. So that's kind of mad. Again, structure discovery and on a virus membrane. It's been one of the uses of STORM. You can do live cell single molecular localization microscopy, but it's not easy. Here's an example of focal adhesions using live cell palm. So if you're doing live cell super resolution, you're almost always going to be using palm because of fluorescent proteins, unless you're doing something on the cell surface and you can use some kind of surface receptor antibodies, etc. But here you can actually see these focal adhesions as the cell is migrating using live cell palm. So what are the limitations of single molecule localization microscopy? Well, the big one is you need to really get your sample preparation right. You need to do very specialized fluorescence labels. You need to make sure you're buffered correctly. If you use the wrong label, it just won't work at all. Some fluorescent proteins just are really difficult to make them blink. Also, the buffers you use and the laser powers that you need to mediate switching events often are really not live cell friendly at all. The buffer is often a redox buffers, so they're controlling oxidation states. Cells really don't like them. And the laser powers can again get very, very intense very quickly. You also need a long acquisition time, right? So if you want the thing you need to do, if you've got very few fluorescent molecules emitting their fluorescence per frame, you need a really large number of frames, right? Because you want to make sure you capture each molecule in the sample at least once. If you don't take enough frames, then you're going to get incomplete structures. You're not going to actually catch the blinking from every single molecule. So you need really long acquisition times. And that's bad for live cell imaging, of course, if you're already using quite high laser intensities. But it's also a bit problematic for dynamic structures. So if you've got a sample that's moving, then, you know, your fluoriform location is going to have moved before you can actually capture it, for example. There's an example of this here. So this is from that same data set as in the last slide, and it's showing that depending on how long you spend, how much time you kind of allocate for a single palm image can actually change what it looks like. So this was the focal adhesions made from 30 seconds worth of imaging. This is what it looked like from 180 seconds worth of imaging for a single frame. So this isn't in a time series, this is just for a single frame, or for 270 seconds worth of imaging. And they're overlaid here in 3 different colours, in cyan, yellow and magenta. And you can see that your structure is going to be different depending on how many frames you acquire to make your single super resolution frame. Okay. And finally, you can get quite bad artifacts when you're trying to localize the molecules. Again, I mentioned briefly, for example, if your blinking is quite dense, so if you're not very sparse, if you occasionally get overlapping fluorophores, that can make the reconstruction go a bit haywire. The algorithms don't really know what to do with those overlapping molecules a lot of the time. Imagine you've got one molecule here, one molecule here. Very easy to localize those as two separate objects. If they overlap, then the algorithm often just thinks it's one big fat molecule, and so you actually lose information. And there's an example of this here. So this is a good single molecule reconstruction of some microtubules. You can see nice, clear individual filaments. On the right, it's the same data set, but analysed differently and analysed in such a way that it's a real problem if there are overlapping molecules. And you can see you get this blurring and this merging of structures, you get incomplete structures, etc. So you can get some really nasty artifacts in single molecule localization microscopy. In terms of manufacturers, all the major, Nikon definitely do a single molecule localization microscope. Zeiss do. There's a company called Oxford Nanoimaging, which does like a little tabletop single molecule localization microscope. And again, pretty easy to build your own in this case because you don't need any additional optics. You just need powerful lasers. So yeah, that's single molecule localization microscopy. So hopefully after that, you're now comfortable with why we can accurately determine the center of a single emitting fluorophore. You know, they always look the same each time, so you can accurately work out where the center is. How you can generate blinking in different ways, and the various advantages and disadvantages of single molecule localization microscopy. This is the one I use the most, so there's a really nice review of it here, but there's also some slides and some demonstrations of the analysis that I've done for other kind of talks that might be useful if you want to look at this further. So that's the main super resolution techniques. I think hopefully one thing that you'll kind of realize from these two talks is there is no one best technique, right? They all have advantages and disadvantages. And for example, single molecule localization microscopy has reliably the highest resolution, but is probably most likely to kill your sample. SIM, on the other hand, you know, doesn't have great spatial resolution, but it's actually pretty straightforward to do. So it's really, what do you want out of your sample? What kind of information are you trying to see? And a lot of the time, diffraction limited is a very good way to go. I know I've kind of come into these talks saying super resolution is great, it lets us fill this kind of gap in resolution between fluorescence and electron microscopy, but all of these methods do have quite strong drawbacks. So if you're struggling to kind of get microscopy working in diffraction limited regime, don't go to super resolution. Okay, super resolution should only really be done once you've optimized and pushed your diffraction limited regime as far as you can take it. If you're going to never go into an imaging problem just straight up super resolution, because you'll cause yourself more problems than you will solve. There are two really nice resources here for kind of comparing different super resolution methods and when you might use them. There's this review in Nature Cell Biology, and there's this really nice poster which actually kind of plots out the advantages and disadvantages and shows you example data sets. Okay, so what's going on in super resolution microscopy at the moment? You know, SIM, STED and single molecule localization microscopy are all pretty established. They've all been used for a long time. Everyone's very familiar with them. So let's think back to the beginning of the last kind of talk. why aren't we just doing electron microscopy, right? That's got really, really high resolution. And the answer is, well, super resolution should work with live cells, right? You know, otherwise you might as well do electron microscopy. So what I'm going to show you here is this is a fluorescence and transmitted light image of some cells that are expressing tubulin and GFP. We've got two interphase cells that are nicely spread and two mitotic cells which are rounded up. And I've got the intensity of the laser illumination up in the corner here. Now what I'm going to do in this movie, I'm just going to increase the intensity of our laser illumination and we're going to see what happens. So increasing the intensity and bad things start happening quite soon. You can see really bad blebbing on these mitotic cells. You can see shrinkage of these interphase cells. And, you know, as we increase that intensity, things get very, very bad for our live cells. And the position where the kind of intensity where this starts happening, where bad things start happening to our cells, is lower than you might think. And even though techniques such as SIM are kind of touted as, you know, SIM in particular, people say, oh, it's very live cell friendly. Out of all the super resolution techniques, it's very live cell friendly. It's actually quite substantially into the regime where you start seeing these bad kind of phototoxicity artifacts. So once again, here is about where you SIM, and that's just when your cells are kind of starting to really show signs of utter distress. This last frame here would be a typical storm imaging intensity, and you can see you just don't want to be doing that with live cells, okay? And, you know, the reason these cells are unhappy is because of phototoxicity. And so there are various things that can cause reasons why light can be phototoxic to cells. So if you're using UV illumination, remember when we were talking about single molecule localization microscopy, you can actually, you know, we needed that for facilitating some of the blinking transitions. You can actually induce an apoptotic ultraviolet response. You can directly damage DNA strands. But the most common reason for phototoxicity in single molecule, well, in super resolution microscopy in general, is that you can generate reactive oxygen species. And that's just a byproduct of exciting lots of fluorescence, and both from the thing you're trying to image and other fluorophores and molecules in your sample. So whenever you're using high intensity illumination, you're generating reactive oxygen species, which then kind of bomb around the cell and oxidize everything and make your cell very unhappy. Okay, so this is a problem, right? We want to be doing live cell super resolution microscopy because, you know, if we didn't care about resolution, we'd use normal fluorescence. If we didn't care about the cell being alive, we'd use electron microscopy. So there are various strategies that we can use to tackle phototoxicity in super resolution microscopy. Obviously, you can try and deliver less light to the sample. So here's just an example of 3 imaging regimes. Wide field, where we illuminate everything in this fat column of light. Confocal, where you illuminate everything along a skinny beam of light. And 2-photon, where you only excite molecules at the focal plane. You can use inhomogeneous illumination to try and reduce the light dose to your sample. For example, multifocal illumination, or delivering your light in kind of temporal chunks. Or you can also do things like spatially adaptive illumination, where depending on what the fluorescence is in your sample, you can either make your light more intense or less intense. So only delivering light to excite molecules where it's needed, for example. And you can also use light sheet illumination regimes. So Gaussian light sheets, for example, you only illuminate the molecules that you're imaging. So again, if you illuminate parallel to your imaging plane, then you get a nice flat plane of light that you can just collect straight in. Or lattice light sheet microscopy, where you can actually illuminate a really skinny plane of light, so you don't even get the problems of illuminating and exciting molecules above and below the focal plane. So we can think about the spatially adaptive techniques, and this is something that's been done a lot in STED microscopy. Beery instruments, for example, have now got commercial systems that can do this. So you have things like rescue STED, where your lasers, in this case, you scan with your confocal spot, your excitation laser, and you only turn on your STED beam when there's fluorescence, right? So you don't want to be actually delivering your STED beam into your sample for places where there aren't fluorescent molecules. You only want to be putting your STED beam on and having it intense where you actually have molecules within your sample. Okay, so this is cool. This uses the excitation laser, scanning through, scanning, oh, there's fluorescence, turn the STED on. Scanning, oh, no fluorescence, turn the STED off. So that reduces the amount of STED light that goes into your sample. And a similar kind of technique is diamond. So this is actually modulating the power of the STED beam, so the intensity of the STED beam, until you either reach some kind of resolution threshold, for example, because remember, let's say we're scanning along and we're like, okay, we found a molecule, we want to try and increase the resolution this much, done it, let's move on. So increasing the power of the STED beam until you've got just enough, because resolution scales with beam intensity instead. Lattice light sheet microscopy is kind of very cool. This is what a lattice light sheet microscope looks like. It looks like not a microscope, and it allows you to illuminate your sample with very thin sheets of light, as shown here, and then detect perpendicular. And this can basically be combined with other wide field techniques. So you can treat each of these sheets of light as a potential plane of imaging for SIM, or single molecule localization microscopy. So that's pretty cool. Less light delivery, not going through the whole sample, but then still doing super resolution within those thin sheets. This is an example of lattice light sheet SIM. This is for actin in a living HeLa cell. So you can see very, very high temporal resolution and kind of very nice spatial resolution as well. And here on the right, we're going to see an example of using lattice light sheet microscopy, the palm. So in this case, it's Dendra2, that photoconvertible fluorescent protein we were talking about earlier, the one that goes from green to red, conjugated to lamin A on the nuclear lamina. And you can see building up this really nice high resolution image of the nuclear lamina. Again, you can do it in 3 dimensions as you take that light sheet through your sample, and you get this absolutely incredibly beautiful 3-dimensional resolution. I'm just going to sit and watch this for a while because I think this is absolutely gorgeous. And again, we're coming back through the nucleus, various slices compared to the kind of diffraction limited on the right. Okay, so lattice light sheet microscopy is pretty cool. It delivers less light to your sample because you don't actually illuminate big fat chunks of the sample, just skinny little sheets. And you can do super resolution imaging within a single sheet of that sample. So SIM has a lot of other developments hardware-wise as well, and this is just an example that I'm using because I think the data is just so beautiful. This is a technique called grazing incident SIM, which again, normal SIM, you'd put your light sheet into your sample, but you'd still get out of focus fluorescence, and you'd get, again, photo damage throughout the kind of column of light as your beam's going through your sample. Grazing incident SIM, again, allows you to restrict your SIM imaging to a thin plane around the focal plane. And again, you can have this all really beautiful, very fast, doubled resolution, multicolour SIM imaging, which is absolutely beautiful for this this modality. And again, really lovely cell dynamics, which are again helped by the fact that you're not damaging your sample as much because you're not inputting light into the whole thickness of the sample. You're just restricting it to a little strip. Play. No. One thing that's quite interesting is the advent of pixel reassignment methods. And these are things like ArieScan and iSim. And people will often say that pixel reassignment methods aren't super resolution methods because you don't really get a huge increase in resolution. You'll get maybe a 1.4-fold increase in resolution compared to SIM, which is 2-fold resolution, and then the much higher resolutions with STED and single molecular localization microscopy. However, these are really popular at the moment because they're not as phototoxic and not as photodamaging. They're also really easy to use. And so if you're thinking about what super resolution technique do I want to use for my imaging, these are often a very good place to start because they can start to tell you, what a small amount of resolution increase can do for your imaging. That might be enough. And it does so without complicating your life to the extent that some of the other techniques do. So here's an example of, I think this is iSim. And you can see you get really nice dynamics in this image. And there's a kind of really lovely review here of current and future developments for SIM in particular, which include a lot of pixel reassignment methods. Play the movie again, why not? Okay, so finally, another way of tackling phototoxicity in super resolution microscopy. I've shown you all these fancy things, this adaptive illumination, this light sheet illumination for reducing the dose of light into your sample. But what if we just turn down the laser intensity? What's the worst that can happen? So this is an example of what would happen for Alexa Fluor 647. So this is some real data, we've got some blinking data, where the incident illumination intensity was about 2 kilowatts per square centimetre. For a kind of comparison, for a benchmark, the illumination of the sun at the Earth, the illumination of the sun at the Earth's surface, it's about 0.1 watts per square centimetre. So this is a high intensity situation. What would this data set look like? Same sample, same imaging, if I just turned down the laser intensity. And this is the super resolution rendering of that data set. Well, it would look like this. So this is at 40 milliwatts per square centimetre. And as you'd expect, it kind of just looks like regular fluorescence imaging. You can't really see that blinking at all. And if I tried to just do something quite silly and just put this data set through my single molecule localization algorithm, trying to find individual molecules, then of course we'll get a terrible image. It just can't find anything that looks like single molecules in that data set. So one thing that you can do is there's a family of kind of computational techniques that actually try and get computational super resolution from low intensity data. One of these is one that was developed in my current lab, Ricardo Enrique's lab, called SURF, which is super resolution radial fluctuations. And this is a method of trying to find the fluorescent molecules without using this kind of binary, is this a molecule? Yes. Is this a molecule? No. These kind of probabilities to say where's it most likely that there are molecules in the image. And for this image here, you can see we actually get back a nice high resolution image without all the missing information in a standard single molecule localization reconstruction. One of the nice things about SURF, for example, is that it can be used with any fluorophore or any microscope. So this is an example of ultrafin GFP image in a confocal microscope. SURF uses 10 to 100 normally frames per single SURF image. So it analyzes the fluorescence in a series of images, compresses it into one in the same way that single molecule localization algorithms do, but without trying to find single molecules. And again, you can see this is live cell data The cell's pretty happy in there, and we've got nice high temporal resolution. Again, the more fluctuations, the more intensity variations, the more blinking you have, the higher resolution you'll get with something like surf. But again, as a starting point, this is quite a good place to start. You're not going to get the highest resolution, but you're going to start seeing where you could potentially go without needing to buy a new microscope or without needing to, do some incredibly fancy sample preparation. And to finish off on the software developments and the cutting edge of super resolution, one thing that's kind of coming to the fore are deep learning based methods. So these use kind of neural networks and powerful kind of artificial intelligence style computing to infer super resolution from maybe substandard data sets. So here's an example of a neural network based technique that uses a neural network to reconstruct sim data. So this is data that was reconstructed with a standard sim reconstruction and same data that went through this SCUNet reconstruction. So looks pretty good. Looks like you've got much more resolution there. And there's other kind of, this is a really, really growing field, but another example is Anapalm. And this learns how to make good super resolution reconstructions with much fewer frames than you'd normally put into a single molecule reconstruction. So this is an example here of a wide field image. If you use 300 frames to make your palm image, then it would look like this. If you use 30,000 frames to make your palm image, it would look like this. And the cool thing with Anapalm is it learns how to make an image that looks like it was acquired with 30,000 frames from data that's only 300 frames. So again, that's pretty cool. A word of warning with anything deep learning is you're never really sure what it's doing, okay? So you could get artifacts and not be aware of them. Artifacts in super resolution microscopy in general, they're present, but you normally know why they're there. In deep learning, you're never really sure if they're there or where they came from. So that's just a word of warning. Okay, so hopefully by the end of that, you're now comfortable with how light can cause damage to living cells and how we can use hardware to minimize phototoxicity in super resolution imaging, so changing the way we deliver light to the sample, and also how we can use software to maybe deal with less good data and try and get more resolution out of that data. And we did a review very recently about phototoxicity in super resolution microscopy, which again goes into much more detail on all of this. Just to wrap up, this is something that you should always bear in mind if you're doing basically any microscopy that isn't just taking an image, if you're doing any kind of processing or any kind of super resolution. So how do you assess your super resolution data and what's it actually telling you? So, for example, what does the intensity mean in a super resolution image? What's the resolution of your image and how do you measure it? And how can you tell if you have artifacts in your image? So be very, very, very careful about inferring intensity-based information like protein counts or, any kind of absolute intensity quantification from super resolution images. Sim and STED intensity should be representative of the true number of molecules, but single molecule localization microscopy intensities can be really misleading. So for example, let's say we've got 2 molecules that we're imaging, and the way we image it, just by a pure chance, our left molecule blinks 3 times more than our right molecule. If you don't analyse that data correctly, or if you don't appreciate what an intensity in a single molecule localization microscopy image means, then you might end up with an image like this, where you say, oh, there's more protein or there's more fluorophore on the left than on the right, when in reality, that molecule just blinked more than the one on the right. So that's something that you need to kind of bear in mind and be really critical when you're looking at super resolution data. Think, you know, what's the author saying this tells us, and is this actually what the information is telling us? And again, the big goal of super resolution microscopy is to increase the resolution as high as possible, but just be wary that high resolution doesn't necessarily equal high quality. So here's a pair of conventional and super resolution images of the same structure. And I made some software a couple of years ago now, I'm still developing a version 2, but it's called Squirrel. It's a method for quantifying errors in super resolution microscopy. And this will give you an error map in this case, saying where the super resolution image is performing well in comparison to a diffraction limited image. You can quantify resolution in various different ways. One popular way, and this is inside Squirrel as well, is Fourier ring correlation, but there's also newer techniques such as image decorrelation microscopy. But what you can also do in Squirrel, for example, is actually measure the resolution in different parts of the image, which is kind of gives you a map that looks like this. So in this, Squirrel is a kind of software tool that can be used to assess the resolution and quality of your images. Cool, great, useful thing, I hope, because it's a shameless plug for my own research. But be really careful about, in your mind, kind of equating high resolution with high quality. In some cases, high resolution imaging is high quality imaging. So if we have a look in this kind of yellow ring, we've got really nice high resolution imaging. That's the wide field equivalent. And our error map isn't really saying that there's anything particularly wrong. But if we look in this grey ring here, you know, that's quite a good resolution, 66 nanometers. That's pretty similar to our resolution in this part of the image. But actually, the intensities have gone pretty mad compared to our wide field image, and it's showing up in the error map. So again, this is more as kind of a kind of lesson in being critical of data. If you're looking at a super resolution paper and they're saying, oh, we've got 30 nanometers resolution, you know, how did they measure that resolution? What's that resolution telling you? And most importantly, that high resolution doesn't mean it's a very high quality. It could do, but that's not necessarily the same thing. Okay, and then on that kind of cautionary note, thank you so much for listening to these seminars. I hope they were interesting and informative, and if not, I hope that at least there are some references that you can go and find more information about the various things that we've talked about, and good luck on your microscopy journeys.